Pal.
Pharmacology of Cystic Fibrosis. Hello everyone, and welcome to our deep dive into cystic fibrosis, or CF for short. Today, we're going to explore the fascinating journey of its discovery, the underlying pathophysiology, epidemiology, and the genetic and molecular basis of this condition. So, let's get started. Cystic fibrosis was first identified in the 1930s, but it wasn't until 1989 that the CF gene was discovered. Researchers like Lap Chi Sui and Francis Collins played crucial roles in identifying the cystic fibrosis transmembrane conductance regulator, or CFTR gene, located on chromosome 7. This gene is vital because it encodes a protein that functions as a chloride channel in epithelial cells. In healthy individuals, the CFTR protein helps regulate the movement of chloride and bicarbonate ions across cell membranes. This process is essential for maintaining the balance of salt and water in various organs, particularly the lungs, pancreas, and sweat glands. However, mutations in the CFTR gene disrupt this function, leading to the hallmark symptoms of CF, such as thick, sticky mucus buildup. Now, let's talk about CF epidemiology. Cystic fibrosis affects approximately 30,000 children and young adults in the United States alone. It occurs in about 1 in every 3,200 live Caucasian births, and there are about 1,000 new cases diagnosed each year. Sadly, half of all individuals with CF live to the age of 31, while the other half do not. Did you know that more than 10 million Americans are carriers of the defective CF gene? This condition is particularly prevalent among individuals of European descent, with an incidence rate between 1 in 3,000 and 1 in 6,000. So, what exactly causes cystic fibrosis? It's an autosomal recessive disorder, which means that a person must inherit 2 copies of the mutated gene, one from each parent, to develop the disease. The most common mutation, known as delta F508, accounts for about 70% of CF cases. This specific mutation results in the loss of the amino acid phenylalanine in the CFTR protein, leading to its misfolding and degradation. When CFTR is dysfunctional, it leads to defective ion transport, causing thick mucus to accumulate in the lungs and other organs. This mucus obstructs airways, promotes bacterial infections, and triggers inflammation, resulting in chronic lung damage. Additionally, the pancreas becomes affected, leading to malabsorption of nutrients due to blocked ducts. Understanding the genetic and molecular basis of CF has paved the way for targeted therapies. These include CFTR modulators, which aim to improve the function of the defective protein. Treatments like ivacaftor and lumacaftor help restore chloride transport and improve lung function, significantly enhancing the quality of life for many patients. The discovery of the CF gene and our growing understanding of its pathophysiology have transformed the landscape of cystic fibrosis treatment. While challenges remain, advancements in research and personalized medicine continue to offer hope for those affected by this condition. Now, let's talk about the pathophysiological abnormalities. In CF, the CFTR protein, which normally helps regulate the movement of chloride and sodium ions in and out of cells, is dysfunctional. This results in reduced airway surface liquid, mucus accumulation, and inflammation, leading to lung damage and infections. So, how do we tackle these issues with medication? Let's explore some commonly used drugs. 1. Bronchodilators. These are medications like albuterol and levolbuterol. They help relax and open the airways, making it easier for patients to breathe. Think of them as the first line of defense against airway constriction. 2. Mucolytics. One key player here is pulmozyme, or Dornase alpha. This drug breaks down the thick mucus, helping to clear it from the lungs. It's like having a little helper to make the mucus less sticky. 3. Anti-inflammatory agents. Corticosteroids and high-dose ibuprofen are used to reduce inflammation in the lungs. Inflammation can lead to further lung damage, so controlling it is crucial. 4. Antibiotics. Patients with CF are prone to lung infections, especially from bacteria like Pseudomonas aeruginosa. Common antibiotics include tobramycin, azithromycin, and caistin, an inhaled form of aztreonam. These help to fight off those pesky infections. 5. CFTR modulators. Now, let's talk about some groundbreaking treatments. Orkambi, lumacaftor/ivacaftor, and Trikafta, elexacaftor/tezacaftor/ivacaftor, are designed to target the underlying cause of CF by improving the function of the defective CFTR protein. These drugs are tailored to specific mutations in the CFTR gene, making them a game changer for many patients. 6. Pancreatic enzymes. Since CF can impair the pancreas, medications like pancrelipase help with digestion by providing the necessary enzymes that the body can't produce adequately. 7. Nutritional supplements. Patients often require a high-fat, high-calorie diet to meet their nutritional needs. Supplements like Aqua ADEKs provide essential vitamins that might be lacking due to malabsorption. Let's take a closer look at some common drug classes and their therapeutic applications. For instance, antibiotics are used to treat bacterial infections. They work by either killing bacteria or inhibiting their growth. Knowing which antibiotic to prescribe depends on the type of bacteria causing the infection. Now let's focus on the cystic fibrosis transmembrane conductance regulator, or CFTR for short. We'll explore its structure, function, cellular processing, the different classes of mutations, and the critical roles of CMP, pKa, calcium, and PKC in its activity. First off, let's talk about what CFTR actually is. CFTR is a protein that acts as a channel on the surface of epithelial cells, which line various organs in our body, including the lungs and pancreas. It primarily functions as a chloride channel, allowing chloride ions to move in and out of cells. This ion transport is crucial for maintaining the balance of salt and water on epithelial surfaces, which keeps mucus thin and helps with proper lung function. Now, CFTR is made up of 5 functional regions, 2 transmembrane domains, 2 nucleotide binding domains, and a regulatory domain. These components work together to ensure that CFTR can open and close effectively, allowing for the transport of chloride ions. Next, let's discuss how CFTR is processed within the cell. After being synthesized in the endoplasmic reticulum, CFTR undergoes several modifications before it reaches the cell surface. This process includes folding and glycosylation, which are essential for its stability and function. If CFTR doesn't fold correctly, it can lead to its degradation instead of reaching the cell membrane, which is a common issue in cystic fibrosis. Now, let's break down the 6 classes of mutations that can occur in the CFTR gene. Understanding these mutations is vital because they determine the severity of cystic fibrosis symptoms. 1. Class 1 mutations. These mutations lead to defective protein production, meaning no functional CFTR is made. 2. Class 2 mutations. Here, the CFTR protein is produced but fails to reach the cell surface due to improper trafficking. 3. Class 3 mutations. These allow CFTR to reach the surface, but the regulation of the channel is defective, preventing it from opening properly. 4. Class 4 mutations. In this case, CFTR reaches the surface but has reduced chloride transport capability. 5. Class 5 mutations. These mutations result in reduced production of functional CFTR, meaning that only a small amount may reach the surface. 6. Class 6 mutations. Finally, these mutations lead to unstable CFTR proteins that have diminished chloride channel activity. Each class of mutation contributes differently to the symptoms and severity of cystic fibrosis, which is why understanding them is crucial for treatment strategies. Now, let's shift gears and talk about the biochemical players that regulate CFTR function: CMP, pKa, calcium, and PKC. CMP, or cyclic adenosine monophosphate, is a signaling molecule that plays a pivotal role in the activation of CFTR. When CMP levels rise, it activates protein kinase A, or pKa. pKa is essential for phosphorylating CFTR, which is a key step in opening the chloride channel. Think of it as a key that unlocks the door for chloride ions to flow through. Calcium also plays a role in this process. It can modulate CFTR activity and help in signaling pathways that affect ion transport. Lastly, we have PKC, or protein kinase C, which enhances the responsiveness of CFTR to pKa. It helps in the phosphorylation process, allowing CFTR to open and close more effectively. Together, these molecules ensure that CFTR functions properly, maintaining the balance of ions and fluids in epithelial tissues. When this regulation is disrupted, it can lead to the thick, sticky mucus characteristic of cystic fibrosis. Pharmacogenetics and pharmacogenomics of CFTR. When CFTR is functioning properly, it helps maintain the balance of salt and water in various organs, especially the lungs and pancreas. But in cystic fibrosis, mutations in the CFTR gene disrupt this process, leading to thick, sticky mucus buildup in the lungs and digestive system. This is where pharmacogenetics comes into play. Pharmacogenetics is the study of how an individual's genetic makeup affects their response to drugs. In the context of CF, this means understanding how different mutations in the CFTR gene can influence the effectiveness of CF treatments. Now, there are over 1,000 mutations identified in the CFTR gene, but not all of them respond to available therapies. This is where pharmacogenomics, which looks at the broader genetic factors influencing drug response, becomes crucial. For example, one of the most common mutations, known as G551D, affects about 4 to 5% of CF patients. This specific mutation can be treated with a drug called ivacaftor, which is a CFTR potentiator. It works by increasing the time that the CFTR channels remain open, allowing more chloride ions to flow in and out of cells. However, not all CF patients have the G551D mutation. Some may have other mutations, like the F508del mutation, which is the most common, affecting around 70% of patients. For these individuals, a combination therapy like Orkambi, which includes lumacaftor and ivacaftor, is used to help correct the misfolding of the CFTR protein. These modulators are designed to improve the function of the defective CFTR protein. But remember, the choice of treatment is highly dependent on the specific mutation a patient has. That's the essence of personalized medicine. Now, let's talk about the importance of genetic testing. By analyzing a patient's specific CFTR mutations, healthcare providers can tailor treatments to ensure the best possible outcomes. This is especially critical for patients from diverse ethnic backgrounds, as they may carry different mutations that don't respond to the standard CFTR modulators. Genetic testing can identify these mutations and help determine the most effective treatment plan. It's a game changer in managing cystic fibrosis. In summary, pharmacogenetics and pharmacogenomics are essential in the treatment of cystic fibrosis. By understanding how genetic variations affect drug responses, we can provide more personalized and effective therapies for patients. To recap, 1. CFTR is crucial for ion transport in epithelial cells. 2. Mutations in CFTR lead to cystic fibrosis symptoms. 3. Pharmacogenetics helps us understand how these mutations affect drug response. 4. Personalized medicine is key to effective treatment strategies. The treatment of cystic fibrosis, or CF for short. We'll now focus on two exciting areas: gene therapy and drugs that target the CFTR protein. Now, let's talk about gene therapy. This approach is particularly promising for CF because it's a monogenic disease, meaning it's caused by a single gene defect. Gene therapy aims to either replace the faulty CFTR gene or introduce a functional copy of it into the epithelial cells of the airways. One method involves using compacted DNA or CRISPR/Cas9 technology. CRISPR is a gene editing tool that allows scientists to cut and correct the defective sequence in the CFTR gene. Imagine it as a pair of molecular scissors that can snip out the bad parts and replace them with good ones. The goal is to restore the function of the CFTR protein, which would significantly improve lung function and overall quality of life for CF patients. Now, let's shift gears and discuss the drugs that target the CFTR protein. There are several classes of medications designed to improve the function of the defective CFTR protein. These include potentiators, correctors, and suppressors. First up, we have potentiators like ivacaftor, also known by its brand name Kalydeco. This drug is designed for patients with specific mutations, such as the G551D mutation. It works by increasing the time that the CFTR channels remain open, allowing more chloride ions to flow in and out of cells. Next, we have correctors like lumacaftor, which is often used in combination with ivacaftor in a drug called Orkambi. Lumacaftor helps the misfolded CFTR protein reach the cell surface, where it can function properly. It targets the most common mutation, F508del, which affects about 90% of CF patients. Lastly, we have suppressors, like atalurin, which are still under investigation. These drugs aim to bypass premature stop codons in the CFTR gene, allowing for the production of a functional protein, even when the gene has mutations that would normally halt protein production. In summary, the landscape of CF treatment is evolving rapidly with gene therapy and targeted drugs. These advancements not only aim to manage symptoms but also address the root cause of the disease. The hope is that with continued research, we can significantly improve the lives of those living with cystic fibrosis. Thanks for joining me today. If you found this video helpful, don't forget to like, subscribe, and hit that notification bell for more updates on health and medicine. If you have any questions or topics you'd like us to cover next, drop a comment below. Until next time, stay healthy.
Pharmacology.